So welcome everyone. And this section we are going to start with Perseus, hand-on training about Perseus. And we have two speakers to help us, to show us how to use the Perseus. And the first speaker is Professor Tami Geiger from Tel Aviv University. She studied biology at Hebrew University of Jerusalem in Israel. And then she completed her master and doctoral degrees over there, under the supervision of the Professor Alexander Levishki. In 2008, she moved to Matthias Mann Lab in Max Planck Institute of Biochemistry. And over there, she specialized in the proteomics technology to apply to the cancer research. In October 2011, she made an exciting move, and then she went back to Israel to have her own research laboratory in the Tel Aviv University. She is mainly focusing on the cancer research and then doing also the cancer biomarker discovery. And the second speaker is Dr. Stefka Tayanova from Roche. She's a senior data scientist over there. And before she joined the Roche, she was working as a postdoctoral fellow in the Jürgens lab in Max Planck Institute of Biochemistry, where she was focusing on the development and implementation of methods and software tools. And before that, she did her master's in Jacobs University, Bremen, in the field of bioinformatics. And then she got her degree in the computational biology from Technical University of Munich, with collaboration of Max Planck Institute of Biochemistry. And so the first speaker, Professor Tami Geiger, here. And if you have any question, all the team members are here to help you, so you could always ask any question during the sessions.
So hi, everyone. Great, some logistics. Okay, so good afternoon. First of all, I want to thank the organizer for inviting me. It's really fun to come here every year. And it's unbelievable that it's already 10 years. And it's also amazing how this changed from the 50 people that we had in Split, Croatia, to this amazing hall. All right, so what we're going to do in the next few hours is go over some basic analysis that are done in Perseus, just very briefly about how we got to this point. So actually, when I joined Matthias's lab, so about 10 years ago, everyone was actually doing their own R code, scripting, whatever, and everyone was a bit struggling with it, and then most of the work was done by bioinformaticians. And at a certain point, we started in discussions with Jürgen. The idea was that the biologists should be able to do their own analysis and learn how to do it without struggling with coding every little thing, and also doing it properly, as you already heard parts of it in Jürgen's talk before, because we don't always get all the proper training in statistics and so on. So now when we use Perseus, we have a huge amount of options. We can do all these different analyses really fast, so we don't have to struggle for one day, run each analysis, but we can just sit for a couple of hours and do a million things. And we also do them properly because the software is already set to do it properly. So the defaults are usually suitable, and you don't have to be a statistician or a bioinformatician to do it properly. And what so I'm a biologist by training, and I'm going to describe you basically a bit from that point of view as a end user rather than the bioinformatician who is programming it, writing it, but basically the one that is actually using it on a daily basis. All right, so what we're going to start with is a data set that I generated many years ago. It was published already in 2011, so nothing very dramatic or exciting, but very basic and very suitable data set to practice on. So this is a data set of breast cancer cell lines, and what I did there is use so it's a SILAC experiment, heavy labeled standard, so everything is quantified relative to the same standard. So we have a heavy label, which is the standard one cell line, and we have the light labels, which are different cell lines. What we're going to do is start looking at the data, examining the data, plotting the data, doing some statistics on it, and basically you can follow in your tutorial basically the whole workflow that we're going to work with, and this is just an example of how to work with such data. So let's start. So the idea is that basically I try to work relatively slow. You should try to follow as much as possible what I'm doing. Try on your own computers. And if you have questions, something gets stuck, raise your hand. There is a whole team of people from Jürgen's group who can help you. And if it's a more major question, fundamental one, then I can stop me and I'll try to answer. Okay. So you can go to your USB stick, and first of all, let's open the software. So you go to Software, Perseus, and you double-click on the program. Okay. So what basically what Jürgen already showed you briefly like an hour ago. So you'll have here a matrix, you'll have here an workflow, and you have some information here. But what is even more important for the beginning is that you have different parts. So you have here the loading on the left side, then all the processing options, which, as you can see, are plenty. So you can really jump. So if you just click on different drop-down menus, you can see that there are really many options of what to do with it. And the idea is that it's so easy and so fast to do it, and you can just play with it afterwards at home and try different things and until you get something meaningful from the data. Then we have all the analysis tools, and we have some additional functions on the right side, which are a bit difficult to see on the screen. So we start by loading the data, okay? So you can see here the green arrow. That's where you actually load. And you have to select a file. And we're going to select a file from the USB stick, go to Data, Practical 2, Breast Cancer. So that's the first data set that we're going to look at today. Now we have to, in this window, you should actually define, so basically you have here, or under normal circumstances, you would have different columns. So this is the column header of the protein groups that you already saw before. And if you want to select it, so you can just select the columns that you want to work on and move them to the right side. And you have to define each of the columns that you want to include as one of 4 options, or 5, mainly 4. So the main ones, these are all your experimental results. So these could be ratios in the case of SILAC, it could be LFQ in the case of label-free analysis, just intensities, and so on. So all the basically experimental quantitative information that you want to see, and in the specific samples that you want to analyze. Then you have the numerical columns. So these are numerical annotations for each protein. So you have, for example, the number of peptides per protein, the number of razor peptides, unique peptides, localization probability if you were talking about a PTM. And then you have the categorical columns, and these would be very often the gene ontology, so the GO categories, KEGG pathways, so all the categories which are string values or strings that describe your protein. In our case, we still didn't load the annotation, so what we have here is basically columns which are only identified by site, reverse, and contaminants. And we will use those to filter the data in the next steps, okay? What you have here in max quant, as you probably already saw, is pluses in the table. And then you have textual annotations, which are basically the G names, uniprot IDs, all the identifiers of each of the proteins. And in some cases, you would also have multi-numerical. So if you have multiple numbers of peptides, for example, per protein or something like that. But this is usually not really used. Okay, so we want to load everything. So we defined all the relevant columns into these groups as one of these groups. And we want to just load it. So you just click OK. And what you will find here is a matrix, okay? And now we can start working, and we can work on the matrix and start applying all kinds of things to the matrix. So the first steps that we're going to do, first few steps, we're going to move from the original data set that we had, the max quant output table, and we are going to convert it to something that we are going to apply all the statistics on. So we're going to do all kinds of normalizations and filtrations and all kinds of modifications. So first of all, we have here the categorical annotations. So we have the reverse proteins. So if I click on the top, you can just sort it. So we have the reverse proteins, we have contaminants, and we have the only identified by site here, true or false, or pluses, different versions. So we want to filter it. So as we just did, we defined it as a categorical annotation, which means that we can now filter it, so filter rows based on categorical column, okay? Because it's a category. We defined it as a category. So for example, we can go now select the column, reverse. When we have a plus, we would tell it to remove the matching rows. Okay, so we want to remove all the rows which have a plus in the column which is called reverse. Okay, now we want to do the same for the only identified by site. So these are proteins that didn't actually go the proper FDR because they were identified by modifications. So I actually do exactly the same, filter rows, only identified by site. If it's true, I want to remove the matching rows. I can do exactly the same if I want to filter out the contaminants. Okay, again, plus and remove. However, I want to show you another option of filtration. And this is manual filtration. For example, in this case, I have all kinds of contaminants, which are keratins, that we often have in mass spec and proteomics data, but in cancer, keratins are actually very important. So I don't want to automatically remove all the keratins. So I can also do it manually. And you can go to the analysis and select rows manually, okay? So this hand. So I click on it. And what you can see here is that I have a second tab. And now I can select specific rows. So I'll sort to have all the contaminants on top. But I don't want to filter all of them. I want to filter whatever. I'll eventually filter all of them. This is just to show you. Okay, so I can, for example, mark these ones. And then I can click here, keep selected rows, which is not what I want, or remove selected rows. So this is just to show the possibility. So I remove the selected rows. Now I will just, to get to get it proper. So I'll go again to the contaminants. And I will remove them. Another way of filtering, for example, I can filter rows based on numerical column, for example. So I can say, okay, I want to filter to keep only the proteins that were identified by more than 2 peptides, for example. So there are all kinds of filtrations that one can do, but we are not going to do at the moment. This is just to show the options. Okay, now our data is actually ratio L to H, ratio H to L. Okay, so the heavy standard versus the experimental system. It doesn't make very much sense when using this SILAC standard, because actually what we are interested in and what reflects the biology is the opposite, is ratio L to H, which means that we want to convert, to inverse the data. So we can very easily do that by basic, transform, and I can do 1 over x. Okay, I can actually transform it in whatever way I want. And since almost everything we do in the log scale, then I can also take these inverted values and log transform them. Okay, I want to stop for 1 second or a few seconds to show you where we are, what it looks like. So we have here the matrix, and you can see that every time I did something, I actually added a new matrix. So I'm not overwriting anything, but I can actually jump between the matrices and go back and see basically the differences between the matrices. I can also go to the workflow here, and what you can see is that all the blue boxes are the different matrices. So I can also jump to matrix 2, and then jump to matrix 4. And I can actually also follow what I did in every step. So for example, I clicked on filter rows based on a categorical column, and I can see here on the right side exactly what I did. Okay, so I filtered reverse, and here only identified by side, and I can follow actually all the steps and go back. If I realize that I made a mistake, I can just go back here and redo it properly. Or I want to do just a parallel analysis, I can also start here and just branch the workflow to as many directions as I want. One other point is that when you look at the matrix itself, you can see the parameters of the matrix. You can see the numbers of main columns. So we have 9 main columns. We have 3 categorical columns, 4 string. We have altogether 9,100 rows, so 9,100 proteins, and so on. So we can see that we started with 9,100. When we started filtering, we got to 8998. Further filtering, we got 89, and so on. So you can really get the information for everything that you did. And this is very convenient for you to monitor everything and go back to places and make sure that you didn't make any mistakes. One more thing that you can do is also export what you did. So any matrix, you can just go to export, which is okay. Here there is an export, which you can probably see better here. You can just export the matrix as a text file, okay? And after doing a lot of processing, you don't want to repeat everything from scratch. You want to upload it again and start from that point. So you can actually call it and exactly know what you did. What you can also do, and this is very useful, is to save the session. So this whole processing that we did here is a session, and you can actually save it and upload it again on the same version of Perseus. So I have to say that this is something that we use really on a daily basis, because my students sit for few days, work on Perseus, and then we want to go and discuss it, and they come with a session, we open it on my computer and see and go over the steps to make sure and help them analyze it in maybe different ways and so on, and check that everything is all right. So this is really very, very useful. And of course, for publication, if you want to go back and you don't remember everything you did, you can always go back to the session and have all this information. Okay. There were questions here? No? So okay.
I asked you to, I can talk about it later.
Okay, so we'll go on. Okay, so now this data set is actually, we loaded here three cell lines, so 3 replicates, 3 cell lines. So we have altogether 9 samples. However, it was a part of a much larger data set of 11 cell lines, so 33 samples. And therefore, there are many columns, many rows that are actually empty. So these are proteins that were found in one of the other 8 that were not included here. And we want to filter it out. We don't want to look, so there are, whatever, 9,000 proteins, but as you can see, looking at the data, that a lot of it is actually empty. So you want to filter it out. So there is a very simple function to do that, and this is filter valid values. So again, filter rows based on valid values. Okay? As you can see, many of them have these small icons that you can also do it faster, but okay, so the idea here is that you want to keep only proteins that have numerical values that were actually measured, that they have an experimental quantitative value in at least a certain percent or a certain number of samples. So for example, if it was not identified at all in any of the 9 samples that we have, of course it's useless. However, if it was only identified or quantified in 1 or 2 of the 9, it's also not very valuable. So you have to set for yourself the threshold that you want to do. I would say that normally we use at least 50% valid values. So something between 50 and 70 or 50 and 80 would be reasonable. So you can filter the number of valid values. So basically by number. So you can say 4 or 5 or 6 in the case of 9 samples. Or you can also do it by percentage, and I would put 70. Then that means that I only retain the proteins that have numerical values in at least 70% of the samples that I have. So all the others, I consider them kind of not reliable enough to actually draw any biological conclusion. So these would all be kicked out. So this is quite harsh, but it's also very important to do, I would say.
Hi. So could you set this percentage or this minimum value per condition?
Okay, so it really depends what you want to do. So usually when I have a whole proteome, so just general proteomics, I would do it on the overall, because I don't expect like all or none issues, cases.
Well, you could have a large number of patients, I don't know, healthy and disease, and maybe you are interested in filtering per number of patients of one of the conditions.
Yeah, so we have a perfect solution for that. This is very relevant to the cases of pull downs, for example, that you have a control and this is empty. Or the best cases are that the controls are empty, or in what your example is that the healthy are, let's say, empty, and the patients have some kind of value. So when the values, the differences are very large. And in this case, you can actually define groups, and Stefka will show you in a bit later in the exercise. She'll show you how to make these groups, but you have the option of selecting it in each group. So for example, I can select 70% in each group. That would be quite harsh. But you can also say in at least one group, which means that if I want it only to be in the patients, in your example, then I would say, okay, I want it to be in at least 70% of the patient, but I don't care how many times I see it in the healthy because I expect it to be very low there, something like that. So you have to think about your own experiment. All the options are here. And they are very useful, and for different experimental designs, different options would be suitable. But you can just select what is suitable in your case. In this type of experiment where there are not huge differences, these are cell lines, so they're kind of a bit similar to each other. There are very few all or none cases, and it's only 3 replicates, so we do it usually as a total. And you can also not only filter by valid values, but you can also filter it by some kind of value, so greater than, whatever, all these different options. Okay? So we do this filtration. I jumped from the wrong place. I'll go back to this and do it again. So I want to do it by percentage, 70. Okay. What you can see also here, that from matrix 7, I had 8774 proteins, and when I filtered relatively harshly, I ended up with 3533. So this was a major filtration because it's this type of data set. So it's usually not that harsh when you actually take the entire data set. Now, just to show you, I made a mistake before. I did it because I clicked on the matrix 3. If I want to clear my, workflow, I can just mark these boxes and I can remove the selected nodes. Okay, so it doesn't bother me later on, so you can clear it every time. If something gets stuck, which I have to say usually doesn't, but depending on the computer might happen, then you can just click on it and tell it to stop activity, and then you're not delayed because of it. But in general, you should also know that we can do multiple things in parallel. So if it's processing one thing and it takes a bit of time, you can also go to a matrix and continue in a different way. So continue things working in parallel, and it doesn't bother at all. Okay, so basically I did this filtration, and now I want to get some kind of an idea of the samples. What I would do is I'll go here to the analysis and the numeric Venn diagram. And I look at all the samples, and I get here a second tab. And it gives me some numerical values or parameters about all the systems that I'm looking at. So for example, after filtration, it tells me the number of proteins that were identified in total in each one of the samples. So this is after filtration. So if I do exactly the same on a different matrix, you would get much more, of course. So we have about 4,000 or 5,000 proteins from each cell line. It can also give you the information about the exclusive, so which proteins or how many proteins were exclusively found in this specific sample. And you can also get the occurrence, so basically 2,100 proteins were not identified in any of the samples, and then it gives you the number, and 2,500 proteins were found in all 9 samples. So you can get this information. And you can also look here at the combinations. So you get all the different combinatorics of these 9 samples. So for example, these 2541 are the ones that occurred in all 9 samples. You can see pluses in all the samples. And it's a bit difficult on this screen. But what you can see, are which proteins these are. So you can get, in this part of the window, you can see the list of protein, and you can export these proteins as well. This list of proteins. Question? Any questions so far? All right. Okay. So now we want to start. Okay, there is one more thing, or two more things that I want to do in order to kind of finalize the preliminary preparation of this matrix. And so I go back to the data, and I go back to the last one. These names are a bit bulky, and it's often very difficult. Until you get to the actual name, you have to read quite a lot, and it's often hidden, like in this case, that you only see the ratio H to L normalized, and this is a repeating unit in all the headers, and we just want to get rid of it. So we want to rename the columns, and you can go to rearrange. So you can see you can rearrange your data in really many different ways. And what we can do is rename the columns by a regular expression, which means that I can remove from all of them the same expression that repeats itself. And what you can do is basically remove the, ratio H to L normalized. Okay, and you can see in the preview, you can see what the names will be. So I actually removed all the repeating parts, and I get a new name. Okay, and now I can actually know which cell line it is. Another option of doing that is to rename columns manually, so not using a regular expression, but just you have here all the columns, and you can just manually change the name to whatever you want. Okay, so that's also applicable, just depending on how much, basically depending on the options that you have, and depending on, it often takes a lot of time to do it if you have many, many samples. So you would want to use the other option. Okay, so now we have these proper names, and the last preparation part that we want to do is add annotations. So we want to enrich the data that we have and take, as Jürgen said, take all kinds of categorical annotations from UniProt, so for example, the gene ontology, KEGG, and so on, and bring them to Perseus. And for that, we actually take the annotation. They always switch places. Annotation columns. Okay, you go to the annotation columns, and you click on Add Annotation. So do you see what I see in your own computers? So I guess for some of you, you don't have all the annotations already configured in the configuration file. So, sorry. Some of you only see this? Yes. Okay, so we want to get all the other options, which are a bit more interesting. So you see here all the different options that we get from UniProt. So how do we get that? So I'll cancel it. So this is just something that we did in advance. But you can actually go to Tools, Annotation Download. And you click on the link, and you actually can select the organism that you want to take the annotation. So Perseus doesn't come with all the annotations because it becomes very, very heavy. So you can select the relevant annotations and load them. You don't actually have to do it now because you have in the USB stick if you go to the USB, you have the data, annotation, and you can select Homo sapiens and keep it in the keep it in this format. And now copy this one and put it in the software. You go to the software, Perseus, bin, conf, annotations, and add it. So you see that I already have it. That's why I saw it before. But you should all copy it to your own computers. Set? Yes. Okay, so I go back to the Perseus window, and I do it again. I click on Add Annotation, and then I would select, for example, the gene ontology and KEG. And move it to the right side. So now I selected those. Note that you actually have to specify your UniProt column because these are matched, these are taken from UniProt and they're matching the UniProt identifiers. So I have to indicate it. Nowadays, when you're using MaxQuant and you take the protein groups file, you will actually have the protein ID would be the UniProt ID, but it's not necessary. So basically, MaxQuant is open, or Perseus is open to working with any other matrix that you want, which is just structured in this specific way. So you don't necessarily have to use UniProt. You just need to match to the UniProt identifiers and get it. And then you can take it from UniProt. In our case, as I told you, this was a bit of an old data, so it's still IPI, which is like ancient history, but seemed suitable for the tutorials. All right, so I click OK. And now what you can see is that I have the GoBP, GoMF, GoCC, and KEG. And each protein has a list of annotations, of different annotations that are relevant to this protein. Okay. So now I'm kind of set with my data set, and I can start working with it. So I filtered it, I added the annotations, and now I can start examining the data itself. And what we're going to do for that is start playing with the analysis tools that Perseus has, which are quite a lot. And these are located here. So for example, the first thing that I want to start doing is make a scatter plot. And I make a scatter plot of the columns. So one column against the other. Sorry. I click this, or visualization, scatter plot. So I want to plot the columns, and I click OK. And as all of these analysis tools, you actually get this second tab, as you already saw, and you get a scatter plot. Okay? And there are many different options here. So I can select what column I want to compare to what column. So for example, I want to compare between replicates, let's say 1 and 3 of cell line HCC 1143. Or I can also compare between different cell lines. Okay, so this is late stage breast cancer versus early stage. Or between normal cells. Okay, so you can see that the correlations are very low when you compare between cell lines, and you can see that the correlations, or just by looking at it, you can also see that the correlations are very high when you compare between biological replicates. Now this is kind of, this is what it looks like, but we also want to know the actual correlation. So you can go here to the correlation tab and click, for example, on the Spearman rank correlation or Pearson correlation. And calculate it. Okay, and now you get that the Spearman rank correlation is 0.91 between the replicates. Now if I take the same as I just did before, and I go between the healthy cell, like kind of healthy cell, and the cancer cells, and I would calculate, I can click here to calculate selected row, then I get a 0.59 correlation. So it's not only that it looks like it's less correlating, but it actually is, surprisingly. Okay, we can also do some more kind of visualization. So we can, here you have the mode button that you can go from zoom. So I can zoom in on a certain area. And I can, so I would zoom out. I can zoom out by double clicking, or I can zoom out by clicking on the full ranges. I can also do a rectangular selection. So for example, select only the top, the most, so these are ratios, so the proteins with the highest ratios. And then you can also get the gene name here, or the gene ID, but the gene name makes more sense. And you can also do the selection with the kind of freehand selection, so not a rectangle, but whatever, any other shape that you want. And what you can do is also export the selection. So for some reason you selected a certain group of proteins, so they were the most abundant, or whatever, you were interested in this group of proteins for some reason. Now you can basically select it. And now we created a new, so you export the selected ones, and you have now a new column which says selection. So these are the proteins. So it's a new matrix, new column, added to this matrix with these few proteins marked. Whatever, 30 proteins marked. So I go back, okay, I selected now randomly, and then I go here to export selection. Points, you go to points, export selection, and you can indicate it in the full matrix. So you saw that we had pluses in the selection, but we had the entire matrix. You can also do export selection and reduce the matrix to create a matrix only with these 30 proteins. Okay, so now we have a new matrix only with the 30 proteins. Okay, now you can also modify it visually. So, okay, I select a group of proteins, and I can change the color. And I can change the shape to, for example, filled circle. And I can change the size. Okay, so now I have these 30 proteins which are very different from the rest, so for some reason. But you can, this is just, these are options that you can play with when you want to make a certain point in your visuals, and it's super easy, actually, to do it. What you can also do is select a certain annotation. For example, you want to see where ribosomal proteins appear in this graph, okay? So you go, before we were in points, now we move to categories. And I sorted by the category name. I would go to ribosomes. Seems a bit tedious, but we'll get there. Okay, so I click on ribosome, for example. So all the ribosomal proteins are here. Note that this is not intensity, this is ratio, so it makes sense that they're not on the high right side. So we have all the ribosomal proteins here, and I want to mark them in green. And I want to mark them as whatever, filled triangles. And okay, so I have all the ribosomal proteins set up here, and you can also do it for multiple annotations. So, for example, whatever, take some random ones, and you can also select them, basically do multiple colors and so on, okay, for each one. Okay, so we looked at this at the mode. What we should also look at the configuration. So this is this Soviet button that you will see all over. So in many properties here, or in many options here, you will see it. And here you can really play with different setups. So you have the grid line. So for example, we want to have the ticks, and we want a horizontal line visible, and you want the major ticks. You can play with the colors or whatever. Okay, so you can actually play with whatever you want here in setting up the graph. And of course, what is very important is the option of exporting your image. So you can actually export it in these different formats. So very often PDF is the preferred one, but you can select whatever you want. So you can export these images and actually use it in publications and so on. Okay. So as you probably already saw in several talks, there are these very nice density plots, and I want to show you how to make them, which is super easy. And we have this density estimation tool, because very often when we have, you have here all the points, and actually you don't know how many points are in the middle because they're just overlapping. So you want to measure of the density of the points in each region. So we have this density estimation, and I would actually calculate the density between replicate, the one that we just looked at, density in the comparison of 1143.1, replicate 1 and replicate 3. And I get a new matrix with a calculation of the density. Okay, so I have the density and I have the excluded fraction. What I'm going to do now is actually use this information, this density information, in the scatter plot that I'm going to remake. Okay, so again, I do exactly the same. I take the scatter plot, I plot the columns, I take the two replicates that I just mentioned, And I'm going to color it here. I'm going to add the density. Okay? So what does it mean? You basically can click here, and you get the color scale, and that tells you basically the density of 1 and the different color scale. What? The mark is just here on the right side. Now, obviously, if I take now and compare to other ones, it would look totally crappy because this is not the density that I calculated. So you have to actually plot the one that you calculated. But you can, of course, calculate the densities of all of them and just select the right one. Okay. So I think these are the main options in the scatter plots. And I want to continue with some fancy stuff. So you can also take, and rather than selecting each time a pair of samples and do the scatter plot, you can also do multi-scatter plot. So you have here a button with all the small rectangles. And I want to plot basically everything against everything. And again, I get a new tab. And you can now see all the different scatter plots. Okay? On this screen, it's a bit difficult to see the axis, but you should be able to see it on your own computers or on bigger screens. You can now also include okay. You can also select any one of the plots, and you will see the plot in the lower right corner. And now what you can do, which is quite nice, is to mark a certain area. So I'm using the exact same tools that we just talked about, but I can select a certain region here, and it will show me where it appears in all other plots. It doesn't give me the option. Never mind. And again, you can export it, and you can play around with it, with the different categories, and so on. I actually want to go back to, I remembered one thing that I forgot in the scatter plots. And this is adding curves to the scatter plot, which could be very nice and useful. So you can go from the data tab to the curves tab here, and you can add an equation. So basically this is y equals x, but you can also add another one, which would be x plus 2. Okay, and you get another line. And you can also do more fancy stuff with polygons and so on, which I'm not going to do for some reason. And you can, of course, remove them and so on. So I can remove it. I can play with it as we just did. So you can really spend the time playing with the different options, and you can see how quick this all is. All right. Another very useful tool that we're going to look at is the histogram. Okay, so we want to see the distribution of the proteins in a histogram. So the ratio distribution that we have. So I just click on histogram again. So visualization, histogram. And I want to prepare histograms of the whole thing. Again, I get a new tab with the histogram. So you can see all the histograms. So we have here the ratio distribution, and the counts. And you can play with the different parameters. So for example, if I want to increase the number of bins, so I can increase it to whatever, 50. I can also change the number of the minimum and maximum values, and so on. So this is very basic stuff, but very fast and easy. And of course, you can also export that. What you can also do, which is very nice, is actually look at the distribution of specific categories. So you have the overall distribution of your proteins, but now you want to focus on a certain group of proteins and check their distribution. So whether they're distributed differently from the rest of the proteins. So I would go again to the ribosomes because they're rather a nicely behaving large group. So now I need to know the ABC, which is a different challenge. Okay, so you can actually see here in a different color, you can see the ribosomes. You can color it, whatever. Now, in many cases, if there are not so many proteins, you want to use a log scale in order to see it better, and you can actually look at it like this. You can just click here on the log, and it kind of pushes everything up. So the difference is, because there are very few, so overall we have thousands of proteins, and then the ribosomes are 100 or 70. So you just want to, this allows you to better see the specific category that you're interested in, and this is very relevant to relatively small categories. So if I look at synaptic vesicle, that would be put on top of the, somewhere in red, which you can hardly see even with the log scale. Okay, so you can see the red one are the synaptic vesicle, in purple or pink are the ribosomes, and you can color it differently. Okay, I'll go back to the kind of normal view. What the histogram is very often useful for is to normalize the data, because in many cases we have some shifts in the data. So when, in principle, I used here the normalized ratio H to L, but sometimes there can be all kinds of, there can be things that actually skew the distribution. And we can normalize it. And as you probably bump to more and more data sets, they don't all look so nice as this one, because they're more complicated, and you might have, for example, in tissues, a lot of extracellular matrix, which will behave totally different from the rest of the distribution, and you want to do all kinds of normalizations. So in order to do that, you have really many different options of normalization. You can look here, normalization by z-scoring, rank, unit vector, blah, blah, blah. What we often do is subtract. We do this normalization on the columns. We want all the columns to look, basically, to be in the same range, let's say, or centered around the 0, which is usually the simplest thing to do. And I want to subtract, for example, the median, or I can subtract the mean. Very often we subtract the most frequent value, so basically the apex of this distribution. And again, I normalize the data, which means that I process the data, I get a new matrix, and now I'll repeat the histograms. and you won't see a big difference because it was already quite nice. But basically, all the highest peak should always be now on the 0 line. So this is a type of normalization. And really, you have different options, and these different options would be suitable for different types of data that you would need, and different quantitative methodologies and different data in general, different samples. How are we with time? 20 minutes? Okay, so one more thing that I want to show is the profile plot. So basically, you can get here the profiles of the different proteins. So you have here the 9 samples that we have, and you have here all the proteins, these 3,000 something. So if I click randomly on a certain protein, you can see the distribution of this protein. So that's kind of nice, but not very useful. But the way we can actually use it is if we have a certain pattern of protein expression profile, I want to look for other proteins that behave, that have the same pattern. So for example, I have, I can take a more extreme one. I can also modify, create kind of a, so I go basically to profiles, and I can create a reference profile, basically. Okay, I can create it manually. So for example, I want proteins that are up in 1143, down in 2218, and then up again. So I can actually draw one manually. So I have the profiles. From the profile, so I have here 4 tabs. I have the profiles tab. That's where I can select the protein. I have the reference profile, okay, that I can create manually. And I can also actually create, basically write the values that I want to have manually. And now what I can, so I can use this and search for proteins that are correlating. So with let's say 10 proteins with a Pearson that are correlating according to Pearson correlation, but can also check, select other ones that are correlating with this specific reference that I used, okay? So I would have here now 10 proteins that behave in a way that correlates with this reference that I had, reference profile. So who are these proteins? I can go here, back to the profiles, and I can bring selection to top. No, I jumped somewhere, sorry. We'll do it again. So basically, you can find, those should be marked here. And then you can, it's a bit jumpy, the mouse here, but you can find them and actually see what these proteins are. What you can also do is, for example, if you select this protein to be your reference, you can go, you want this protein to be your reference, you can go to the references and make the reference from the selected protein. So you selected a specific one. So your reference, you want your reference to be whatever. MVP, or no, CNIH4 for some reason. And you move and you define it as your reference. And now you will look for all the proteins that are correlating with that specific protein. Or you can also look at categories and select the proteins that are, so you can select a category randomly, I didn't even see what it is. Protein tetramerization. And now you can create a profile from the average of these proteins, okay? Which is a very interesting profile. Okay, so you can play with it and really get all kinds of proteins associations that you would not necessarily see in any other way. You can also, I find it very useful if you have some other parameters of the systems that you want to include. And you can check, for example, the proteins that correlate with patient survival, for example. So you can actually create a fake profile, which is the patient survival, and then look at what is correlating with it, and so on. So you can use it in various ways. And again, you can export the selected ones in different ways. You can export the image, as in every case so far. Any questions up to this point? You're very quiet. Stefka, how do you want to go? Do you want to take over? Do you want to? So basically, the next thing that we want to move to is more of the statistical analysis that Stefka is supposed to do. And we'll show you some more.
Hi, thank you. I have one question. Like now I have 19 metrics. If I did something wrong, like 7 or 9, can I automatically change there without deleting after all of them after 9?
So if you found that you did something wrong, so now you're in matrix whatever.
Yeah. Do I need to delete all of them and start over, or I can?
And now you want to start again from 6?
Yeah, like 6. I did something wrong. Can I just only change here?
No, you have to redo
everything again. Okay.
Yes, but you'll do it much faster.
Yeah, for sure. And the other thing is for the land, the different like how to say
the profiles.
Yeah. The profiles. There's one FDR.
Yes. So it's related to the correlation. So if you should apply FDR filtration to get the ones that are significantly correlating with your profile.
Oh yeah, but if I clicked FDR, nothing changed. So I don't know how to do that.
Yes, buttons not working is not my territory. It's where is I guess someone from Jürgen's group should, or Jürgen, I don't know.
No comment.
What?
I don't know.
No, there was a question here.
Yeah, there were a lot more questions, so I think here. Yes, you want to ask a question?
When I save the SPS file, then when I will log them, open it again, all the graphics will be maintained or not? Or is there a way? Because I'm not very...
The graphics is basically maintained, but some of the conversions of colors, I think it depends a bit on the version. I think some of it doesn't. and some does. So you'll still have the scatter plot as you had it, but you maybe will lose some of the coloring.
Hi, is there a possibility to save all those like basic steps as a template so that if I have a new data set, which is basically the similar or same experiment, that I can process it with this template?
I think it continues to, what, Jan? Yes? It has
so
been on
at
the to-do list for the last 10 years.
Yeah,
So
so we have an internal version that partially does that, and we mainly use it for testing, but it's actually really difficult to do this in a good way, because if, for example, the column names of your two data sets are different, then how would you propose Perseus to know what it has to do? So at the moment, you will have to always do it manually.
Hi. Yeah. So it's more a question to the
where?
Me, but it's not a question to you really, because it's about the Perseus versions. So the thing is that the SPS files from different versions cannot be opened in new versions. Is there any possibility that you could allow for downloading the previous versions of Perseus as well, so that if we want to recheck an analysis we did like 5 years ago, we can still do that?
Yeah, so we get these requests a lot. The main issue is that we do not want to support old versions, because we fix bugs all the time, and the new versions should hopefully always be better than the old versions. So for now, we encourage you to kind of save the old versions if you're eager to reopen the sessions. Sometimes you can open them with newer versions, and if you really kind of lost the Perseus version, then the mailing list usually is a good place to ask.
Okay. Yeah, because it was mainly because if you cannot use it. So if you are able to open it anyhow, then you can at least export it and then import it back to the new versions to kind of like redo whatever, but just to check what you did or something like that, it would be really useful. But yeah.
Yeah, so we usually just keep many versions. And we just go back to the one, and we write on the session name the version that it was used, and that works quite well.
So I think the new version of Perseus will also kind of say which version you should have used to open it if it fails.
Just a silly thing, I think, but is there any way to, in the multi-scatter plot, to plot the R values if you calculate some correlation or something?
Yes, you can do it for all of them together, basically. It somehow, we didn't see it here, but you can do all the statistics you can do on all the plots.
So if you make a heat map, can you export it? Does the intensity gradient also?
Louder a bit.
Ah, sorry. If you make a heat map.
Heat map is Stefka later. Yes.
There are some kind of gradient, like the scatter plot.
Yes.
Will you, and you export the figure? Will you also export the gradient, the colors?
The colors, the color bar.
The color bar, yeah, exactly.
I think so, and if not, you just copy it. No, we probably just copy it. That's it. So, what? So we'll have the break now for half an hour, and then Stefka will continue with the second part.